Hi, welcome everyone. This is Vural Tagal, and I'm from Moffitt Center in a Department of Integrated Mathematical Oncology. Today, I will talk about our research, which is focused on whole genome doubling. And in this particular study, we discovered that triple negative breast cancers with low ploidy are hypersensitive to gemcitabine treatments. So because of the time limitations, I will be brief on some slides, but in this project, in this study, we basically to understand the differential response of cancer cells in a ploidy dependent manner, and we developed a three-tier framework consisting of computational, in vitro, and in silico components to understand this phenomenon of ploidy and drug response. So before starting the research and data, I would like to briefly inform on some particular concepts that will be useful to understand the results. And here on the left side, very simply, you see that the fate of a cell, a growing cell, is two right now. One of them is, as we all know, mostly mitotic. If the cell goes through the mitotic cell cycle, so two daughter cells are formed. But emerging data recently show that cells under antimitotic treatments or some stress conditions can go through an alternative cell cycle through polyploidy, and when they go through these polyploid cycles, they can first continue the replication of DNA, but omit mitosis to form these enlarged and polyploid giant cancer cells. Then later, these cells can form multinucleated or mononucleated cancer cells. And after the therapy or stress conditions, actually it was shown that these cells can re-enter the proliferative cycle and grow in numbers again. So more importantly, this mechanism introduces drug resistance. For that reason, it became one of the important topics for drug or cancer medicine. So, and in the lab, again, we are focusing on the whole genome doubling event. And in parallel to this drug discovery research section, we also try to understand the very basic fundamentals of the needs of cancers with differing ploidy. So, and here throughout the presentation as well, will indicate one set of chromosomes, just to refresh the memories or introduce few important terminology. And 2N is the normal diploid or DNA content for a diploid cell, whereas 4N is a tetraploid cell. So in very basic hypothetical thinking, we thought that cells with hyperploidy will need more amount of nucleotides as well as any other building blocks just to continue to grow and divide. And on the right side, you simply see that how the nucleotide biosynthesis occur. And according to, again, our hypothesis, normally 4N cells should produce more nucleotides to support the double amount of genome and replication. And in parallel to the same idea, then we thought that if that is true, then these cells, or at least cancers with differing diploidy, should show different responses to the inhibitors of these nucleotide biosynthesis pathway. And actually, there are multiple drugs already in clinic that are used as chemotherapies for cancer. So we tested this hypothesis on one of the isogenic triple negative breast cancer cell lines that has near diploid and near tetraploid genome. And this is again matched pair of the same cell line. And among the tested 6 compounds, here you see that on the left, only cells show differential response to gemcitabine. And we perform similar viability assays or orthogonal viability assays with crystal violet. Here on the, again, left, you see that SAM159 cells with diploid genome showed hypersensitivity, whereas when the same cells were tetraploidized, become much more tolerant to the drug. And at higher concentrations, the cell growth is not affected as much as the diploid parental cell line. However, this was not the case when the same cells were treated with other chemotherapy agents in the panel. So in addition to this one, isogenic pair, we also obtained another triple negative breast cancer line, MDA-MB231, from our collaborator, actually, Marusik, from the Marusik lab. And we performed the assay and we observed that similarly to the SAM159s, MDA-MB231 cells, when tetraploidized, become much more tolerant to gemcitabine therapy. And lastly, on the right, you see that our search for our analysis of the public databases, which are already showing the dose response curves for multiple drugs, including gemcitabine, and breast cancers from breast cancer cell lines show hypersensitivity to a group of compounds or drugs, and gemcitabine is one of the most cytotoxic one, and confirming our observations already coming from independent resources. And we published this earlier in the past as well, coming from one study that was investigating, again, the effect of Ploidy. So to understand what kind of growth the cells adopt under the treatment of gemcitabine, we utilized our latest computational solution, Ploidy, and very simply, Ploidy takes bright field images, bright field microscopy images, and segment cells, counts the number of cells, and measures the cell size to inform whether they go into the polyploid cycle and become polyploid giant cancer cells or not. And our analysis here on the right show that under the treatment, 2N or diploid cells, you can see the majority of cells already dead, but tetraploid SAM159 cells actually grow in size and then become polyploid. So to confirm the Ploidy results, we also performed another assay with microscopy. And on here on the left side, you see the results with the diploid SAM159s, and on the right with the tetraploid cells. So bottom row shows the cells after 4 days of gemcitabine treatment. And confirming the previous observations of ours, you can see here that diploid SAM159 cells hypersensitive to the treatment, whereas the tetraploid cells actually now look larger, and nuclear size, here you can see it with the nuclear reporter marker and MCHERI is larger. And this shows that in the basically after the treatment, SAM159 cells grow into polyploid giant cancer cell state, and then that is how they tolerate the treatment. So in the third part, we wanted to create a mathematical model and wanted to inform the clinical decisions for both precision medicine according to the ploidy of cancer cell, and also we would like to accurately predict the dose that has to be used. So here you see the simple model that took the information from our like in vitro studies, and in this model we basically introduce the polyploid cell numbers and cell death under treatment, cell proliferation, and then in one part we also add the depolyploidization of these polyploid giant cancer cells after the treatment. But here we have the simplest summary of it. And then basically after we performed lifestyle imaging to understand again the parameters and give the parameters to our model, we perform this lifestyle imaging and check the cells under different concentrations of gemcitabine and treated them for 9 days. So here you will see the summary of basically how the cells behave. And in these two videos, you will see the cells with the red cherry reporter. And if the cell that starts, cytox dip red, which will show up in blue to inform the for the cell that counting. So after starting the treatment in a very short time, here basically the video now will show that just the diploid cells on the left already responded to the treatment very dramatically, and almost all of the cells died. However, the tetraploid SUM159 cells first grew in size, and then after the 5th day, actually the cells could re-enter the proliferative cycle and continue to grow back. Yeah. So as a summary of the whole model flow chart, after performing this time-lapse lifestyle imaging and making the quantification of important parameters, we introduce those values. Here in the second line, you see how the cells respond at different concentrations of gemcitabine and what the cell numbers are. We introduced the parameters to the model, and at the end, basically the whole model in this particular setting informed us that tetraploid or cancer cells, triple negative breast cancer cells with tetraploid genomic content needs about 4 times more gemcitabine treatment to be responsive. So lastly, we wanted to understand how these cells actually tolerate the treatments with gemcitabine and understand the mode of action that takes them safely out of the mitotic cycle and grow into polyploid giant cancer cell state. So already it is actually known that gemcitabine causes replication fork or DNA replication stress, and that can cause the stalled replication fork. And if the stress continues in a prolonged time frame, the open DNA strands start to accumulate DNA damage response components that introduce the cell that there is DNA damage. And eventually, accumulation of this DNA damage response creates cell death. So we tested this hypothesis, and we wanted to see if this is the toleration, differential toleration of these breast cancer cells to gemcitabine. We use the commercially available drug candidate for the DNA damage response inhibition, and the PF 473, 766, 473, 76. And then in actually both cancer cells, in very short time, we observed that cells were not able to enter anymore the polyploid giant cancer cell state and immediately die. So in summary, we, with this study, identified that under the treatment or with gemcitabine treatment, cells exit in the tetraploid breast cancer line, the mitotic cycle, and go into the endoreplication with only growth and synthesis, DNA synthesis phases, and then continue to grow safely in that new cell, alternative cell cycle until the end of the treatment. And when the therapy is terminated, cells can get back to the proliferative cell cycle and grow in numbers. And among all the tested compounds here and drugs on the right side, you see only gemcitabine showed this differential effect. So in conclusion, gemcitabine treatments basically caused differential cytotoxicity on triple negative cancer cell lines with low Ploidy, and clone ID could successfully identified the polyploid giant cancer cell state, and our mathematical model has the potential to inform the clinic for the correct drug selection and dose optimization. And finally, we would like to ultimately create this therapy duets, Ploidy and drug, to actually inform precision medicine further by simply checking the ploidy of the cancer cells and then informing the drug decision. So I would like to thank all our collaborators and lab members here, especially, again, Henri, for the cell lines. And lastly, if you would like to be a part of Moffitt Cancer Center and become a PhD student, so you are welcome to join us. And the application deadline is December 1st every year in the coming cycle. And now I can take maybe some questions, or if not, we can talk later after the
We're running out of time, so if you have questions, please speak to Vural.